friends, in today's lecture, we will talk about DNA repair mechanisms and pathways. DNA is the basic unit of inheritance and maintains the integrity and function of living organisms. However, it is constantly exposed to damaging agents which can cause DNA damage. These agents include radiations like X-rays and UV rays and many chemical agents like alkylating agents, hydrocarbons and anti-tumor agents. Additionally, errors can occur during DNA replication leading to harmful mutations. To overcome the harmful effects of DNA damage, cells have various DNA repair mechanisms which cope up with the damage and errors and repair the DNA. Although all cells possess a large number of different types of DNA repair mechanisms, each is relatively specific for a certain type of DNA damage. These repair systems can be grouped into two main categories: direct repair and excision repair pathways. Direct repair pathways include photoreactivation, repair by ADA protein, and repair by ALK-B protein. Excision repair pathways include nucleotide excision repair pathway, base excision repair pathway, mismatch repair pathway, and the SOS response or SOS repair. Although it might seem that direct reversal of damage would be the simplest way to correct the damage, in most cases, the reverse reaction is not possible for thermodynamic or kinetic reasons. In a few cases, the reaction is reversible, and in some of these cases, mechanisms have been developed to take advantage of that reversibility. Still, there are very few examples of direct DNA repair. Albert Kellner was trying to measure the effect of temperature on repair of ultraviolet damage to DNA in the bacterium Streptomyces. However, he noticed that damage was repaired much faster in some bacterial spores than in others kept at the same temperature. Obviously, some other factor than temperature was operating. Finally, Keller noticed that spores whose damage was repaired fastest were the ones kept most directly exposed to light from a laboratory window. When he performed control experiments with spores kept in the dark, he could detect no repair at all. Thus, he realized that light played a major factor in DNA repair. The light repair came to be known as photoreactivation and was the first DNA repair process to be discovered. Let us understand the details of this repair process. This is a DNA duplex. When exposed to UV radiations, it leads to pyrimidine dimer formation between two adjacent thymine moieties. We have already seen the effect of UV radiations in one of our previous modules. You can visit the channel website and look up for this video on physical agents or physical mutagens. It is catalyzed by an enzyme called photolyase. This photoreactivation repair process is catalyzed by an enzyme called DNA photolyase, and it is mainly responsible for the repair of these pyrimidine dimers in DNA which are caused due to the exposure to UV radiations. This enzyme can actually bind DNA even in the dark. It scans the DNA, and wherever it comes across a distortion due to pyrimidine dimer, it will stop there and it will bind to it. When exposed to light, it gets energized by the energy derived from the visible light and utilizes this energy of the photons to break the cyclobutyl ring structure between the two pyrimidine dimers. Thus, the damage is repaired and the photolyase is finally released. This enzyme photolyase is actually made of two subunits which are coded by the two genes PHRA and PHRB. Photolyze contains two chromophores responsible for absorbing the light energy or the energy of the photons. In all such photolysis, one of the chromophores is methylene tetrahydrofolate or 8-hydroxy-5-deazacflavin and the other is FADH. Photolyze can bind to DNA at any region and it then scans the DNA and stops wherever it comes across a distortion due to the pyrimidine dimers. The enzyme binds to the thymine dimer and the first chromophore folate is used to harvest the light energy. This energy is quickly transduced to FADH, making it FADH2. The now electronically excited FADH2 can transfer the high energy electron to the dimer, causing the cyclobutyl ring to break and separate the two thymine molecules again. However, the distribution of photolyze is not universal. Photolyses are found in bacteria, fungi, plants and many vertebrates, but not in placental mammals. It is widely present in microorganisms, but it is absent in Bacillus subtilis. It is present in Saccharomyces cerevisiae, but absent in Saccharomyces pombe. In addition, 6,4 photolyses have been found in insects, reptiles, and amphibians, but not in E. coli, yeast, or mammals. So let us quickly revise this light repair mechanism or photoreactivation. As you can see, this is a DNA duplex. Due to the exposure to UV radiations, pyrimidine dimer or thymine dimerization has taken place between two adjacent thymines here. Remember, the ring formation takes place between the fifth and the sixth carbons of the thymines to form the cyclobutyl ring. This enzyme photolyase now scans the DNA, binds and scans the DNA, and when it comes across this thymine dimer, it halts here. When exposed to light, this photolyase is activated and it absorbs the light of the photons. And as we have already seen the mechanism, it is responsible for breaking this cyclobutyl ring between the thymine dimers. And when it has broken this ring between the thymine dimers, the enzyme is then released and the DNA damage is thus repaired. Let us now move on and talk about another direct DNA repair example, that is direct repair of alkylated guanine. If you remember from our module on chemical mutagens, the guanine is alkylated at O6 by alkylating agents like ethyl methane sulfonate and methyl methane sulfonate. An enzyme called O6 alkyl guanine DNA alkyl transferase is responsible for the repair of O6 alkyl guanine. This enzyme is also called ADA protein encoded by ADA gene in E. coli. It is made up of 354 amino acids and at the 321st position, a cysteine residue is present. The guanine in this DNA duplex has been methylated to form O6-methyl guanine. DNA methyl transferase or ADA protein approaches the site. It binds to the damaged DNA site and transfers the alkyl group of the alkylated guanine to the sulfhydryl group of the 321st cysteine residue. Thus, the enzyme now becomes alkylated and the damage is repaired. But the protein loses its activity and hence this enzyme is not an enzyme in the correct or the strict sense. Unfortunately, once the alkyl group has been transferred to the protein is permanently inactivated because alkyl cysteine is very stable. Additional repair requires the synthesis of new protein molecules. To help accomplish this in E. coli and perhaps in other organisms, the alkylated version of the protein induces increased transcription of the gene encoding the protein. The alkyl groups handled by this protein range in size from methyl to benzyl. Homologs of these enzyme or protein are present in all living organisms. A third example of direct DNA repair is direct repair of 3-methyl cytosine. This is brought about by ALK-B, a protein that is found in most, perhaps all, living organisms, including humans. This protein is a member of enzymes called alpha ketoglutarate dependent and iron dependent oxygenases, which use iron oxo intermediates to oxidize chemically inert compounds. In the process, alpha ketoglutarate is converted to succinate and carbon dioxide, which is similar to one of the steps in the tricarboxylic acid cycle. ALK-B is capable of reversing methylation at position 1 of adenine and structurally similar position 3 of cytosine, as can be seen on your screen. The reaction catalyzed by ALK-B couples the oxidative decarboxylation of alpha-ketoglutarate acid. This leads to the hydroxylation of the methyl group. The methyl group then spontaneously decomposes to formaldehyde, restoring cytosine. Thus, the original base is restored and the damage is repaired. A final example of direct damage reversal is the sealing of a subset of nicks in DNA by DNA ligases. However, DNA ligases can only seal nicks between 5' phosphates and 3' hydroxyls. Nicks with other configurations or nicks accompanied by additional backbone or base damage require more complicated processing prior to repair and would not be classified as direct damage reversal mechanisms. We talked about direct DNA repair, where we discussed photoreactivation, DNA repair by ADA protein, and DNA repair by ALK-B protein. In today's lecture, we will talk about the excision repair pathways, and we shall be talking about the excision repair pathways like NER, BER, MMR, and SOS. Major DNA repair mechanisms take advantage of the facts that DNA is double-stranded and the same information is present in both strands. All excision repair pathways follow a few common steps. The first step is recognition of the damage when the damage is present in just one strand. The second step is removal or excision of the nucleotide along with the damage. The third step is new DNA synthesis using the complementary strand as template, and this is followed by ligation where the nick is sealed. All organisms, prokaryotic and eukaryotic, employ at least 4 excision repair mechanisms. These include nucleotide excision repair mechanism, base excision repair, mismatch repair, and SOS repair pathways. We will be talking about these pathways in the coming lectures. Let us first today talk about nucleotide excision repair. The huge variety of DNA reactive chemicals in our environment, combined with the huge variety of alterations that can be produced by radiation and by oxidative and free radical attack on DNA, can generate so many types of damage that coping with all types of damage by evolutionary development of damage specific DNA enzymes would be difficult, if not possible. Fortunately, a different, more flexible damage repair mechanism has evolved in living organisms, which is called the nucleotide excision repair mechanism. This recognizes damaged regions based on their abnormal structure as well as on their abnormal chemistry, then excises and replaces them. NER particularly is responsible for repairing the pyrimidine dimers that are formed in the DNA when it is exposed to UV radiations. In all organisms, NER involves the following few steps. The first is damage recognition. The second is binding of multiprotein complex at the damaged site, followed by double incision of the damaged strand several nucleotides away from the damaged site on both the 5' and 3' sites. This is followed by removal of the damage containing oligonucleotide from between the two nicks, followed by repair synthesis by a DNA polymerase, and at the end there is ligation by DNA ligase. Let us first discuss in detail the NER in prokaryotes. In prokaryotes, nucleotide excision repair typically handles bulky damage that distorts DNA double helix. NER in E. coli begins when damaged DNA is clipped by an endonuclease on either side of the lesion, sites 12 to 13 nucleotide apart. The pathway allows damaged DNA to be removed as part of resulting 12 to 13 base nucleotides. The excision is achieved by action of three proteins UVRA, UVRB and UVRC coded by the genes UVRA, UVRB and UVRC respectively. UVRB cannot bind to DNA all by itself. It is helped by UVRA. The first thing that happens in the pathway is that two molecules of UVRA combine with one molecule of UVRB in an ATP-dependent reaction to form UVRA2B complex. This UVRA2B complex then binds to the DNA. The complex recognizes the damaged DNA by the distortion, but it can also bind to undamaged DNA and then slide to the damaged DNA binding site. The binding of the complex to the damaged DNA unwinds a 5 base pair region around the lesion and kinks the DNA around it by about 130 degrees. UVRB has a hydrophobic core. UVRB uses the energy of ATP to flip the unbound bases to face the core. As a result of this, a tight hydrophobic bond forms between UVRB and DNA. The formation of tight bonds between UVRB and DNA weakens the contacts between UVRA and UVRB. This causes UVRA to dissociate, leaving behind a stable UVRB DNA complex. Another ATP is hydrolyzed at this step. The dissociation of UVRA is essential because it triggers the binding of UVRC to the site. UVRB and UVRC are endonucleases responsible for the excisions. UVRB makes a cut approximately 4 nucleotides 3' to the damaged site. This causes the DNA to straighten up. This conformational change causes UVRC to bring about a 5' incision approximately 7 nucleotides away from the damage. Thus, a 12 to 13 oligonucleotide is excised. The binding of UVRC is weak and it slowly dissociates. The dissociation is hastened by UVRD protein, which actually is a helicase. It binds to the nicks and uses the energy of hydrolysis of ATP to displace UVRC. The dissociation of UVRC leaves the 5' incision free for DNA polymerase 1 to bind. Binding of DNA polymerase 1 displaces UVRB along with the excised oligomer. DNA pol 1 synthesizes the 12 to 13 nucleotide long patch, and finally ligases is recruited to seal the nick. Let us quickly revise what we have just seen. This is a double-stranded DNA which has been formed a thymine dimer because it was exposed to the UV radiations. Like I told you in the previous slide, UVRA2B complex is formed It binds to the DNA, scans it, and wherever it comes across a pyrimidine dimer, it stops there. Energy from ATP is used to bend the DNA and change conformation of the UVRB protein. And as a result of the ATP hydrolysis, the UVRA dimer is released. At this point, there is a strong bond formed now between UVRB and the DNA. And the dissociation of UVRA here triggers the binding of UVRC. UVRB brings about a 3' nick and UVRC brings about a 5' nick. The binding of UVRC is weak, and as a result of that, it dissociates from the site, and UVRD, which is a helicase, it binds and it causes the oligonucleotide to be released from here. This leads to the formation of a gap here, and at this gap to the 3' OH end will bind the DNA polymerase 1. It will synthesize and fill this gap by reading the complementary nucleotides on the template DNA on the intact strand, and as a result of that, it will fill this gap. Finally, a DNA ligase is recruited which will seal the nick, and in this manner, the pyrimidine dimer damage which was formed here has been repaired. In prokaryotes, it has been found that the transcribed DNA is repaired faster. It leads to what is called as transcription coupled NER. When RNA polymerase comes across a bulky lesion, it stalls. This stalling is a signal for the transcription repair coupling factor to bind to the site. The binding of TRCF results in two events which take place simultaneously. One is dissociation of the complex of RNA polymerase and RNA transcript. The second is simultaneous recruitment of UVRA2B complex to the site. Once the UVRA2B is recruited to the damaged site, the pathway follows as we have discussed in the previous slide. This system ensures that the coding genes are repaired faster on a priority basis. Nucleotide excision repair in eukaryotes shares the same general plan as that of prokaryotes. However, it is more complex and involves more number of proteins as compared to prokaryotes. The proteins which are involved are encoded by genes which are present on an XP gene cluster containing 8 genes: XPA, XPB, XPC, XPD, XPE, XPF, XPG, and XPV. Humans who have impaired XP proteins have a non-functional NER and cannot repair pyrimidine dimers. This leads to xeroderma pigmentosum, that is the skin cancer. NER takes two forms in eukaryotes. It can involve all lesions throughout the genome, and hence it is called the global genome NER, or it can be confined to the transcribed strands in genetically active regions of the genome, as we saw also for the prokaryotes, and it is called the transcription coupled NER. The mechanisms of these two forms of NER share many common aspects, but the method of recognition of the damage obviously is different. So let us first discuss the global genome NER. The first step in human global genome NER is the recognition of the distortion in the double helix which is caused by DNA damage. This is where the first XP protein, that is XPC, gets involved. XPC together with another protein HHR23B recognizes a lesion in the DNA, binds to it, and causes melting of a small DNA region around the damage. XPA, which has an affinity for damaged DNA, is also involved in an early stage of damage recognition. XPA binds to many of the other factors involved in NER, so it may verify the presence of a DNA lesion in DNA that is already denatured and helps to recruit the other NER factors. Two of the other XP genes, XPB and XPD, code for two subunits of the general transcription factor TF2H, implicating this general transcription factor's importance in NER. These two polypeptides have the DNA helicase activity. So, TF2H is responsible in enlarging the region of melted DNA around the damage. The fact that TF2H interacts with a number of other NER factors suggests that it serves as an organizer of the NER complex. The melting of DNA by TF2H attracts nucleases that nick one strand on either side of the damage, excising a 24 to 32 nucleotide oligonucleotide sequence that contains the damage. Two exonucleases make the cuts on either side of the damaged DNA. One is the XPG product, which cuts on the 3' side of the damage. The other is a complex composed of a protein called ERCC1 plus XPF product, which cuts on the 5' side. These nucleases are ideally suited for their task. They specifically cut DNA at the junction between double-stranded DNA and single-stranded DNA created by the TF2H around the damage. Another protein known as RPA helps position the two exonucleases for proper cleavage. RPA is a single-strand binding protein that binds preferentially to the undamaged strand across the lesion. The side of RPA facing toward the 3' end of this DNA strand binds the ERCC1 XPF complex, and the other side of RPA binds XPG. This automatically puts the two exonucleases on correct sides of the lesion. Once the defective DNA is removed, DNA polymerase epsilon or delta fills in the gap, and DNA ligase seals the remaining nick. The role of XPE is not very clear yet. We have already talked about transcription coupled NER in prokaryotes. Let us now see this mechanism in eukaryotes. Transcription coupled NER uses all of the same factors as does global genome NER, except for XPC, because XPC appears to be responsible for initial damage recognition and limited DNA melting in global genome NER. This role in transcription coupled NER is played by RNA polymerase. When RNA polymerase encounters a distortion of the double helix caused by DNA damage, it stalls. This places the bubble of the melted DNA, which is created by the polymerase, at the site of the lesion. At that point, CSB protein binds to DNA. This leads to the recruitment of other factors. RNA polymerase, along with the transcript, is removed. From that point on, these factors would behave much as they do in global genome NER, enlarging the melted region, clipping the DNA in two places, and removing the piece of oligonucleotide DNA containing the lesion. RNA polymerase plays the role of damage detector. It is constantly scanning the genome as it transcribes, and lesions block its passage, demanding attention. Lesions in parts of the DNA that are not transcribed would not be detected this way, but they can wait longer to be repaired because they are not blocking gene expression. Thus, the fact that non-coding lesions such as pyrimidine dimers and 3-methyladenine block transcription as well as DNA replication is useful to the cell in that these lesions stall the transcribing polymerase, which recruits the repair machinery, and the lesion is repaired on a priority basis. We have discussed the first excision repair pathway, that is nucleotide excision repair, where we have seen this repair pathway both in prokaryotes as well as in eukaryotes. We will talk about yet another excision repair pathway, that is the base excision repair pathway, the pathway most commonly employed to remove non-bulky lesions, particularly Incorrect bases like uracil or damaged bases like 3-methyladenine is called the base excision repair. Actually, it's misleading to talk about this as a pathway because there are numerous variations, each specific for a different type of incorrect base. Nevertheless, all of the variant pathways have features in common, and each of the pathways can be considered to consist of 3 steps, with the steps 2 and 3 being common for all the pathways. This is a double-stranded DNA with a damaged base. The first step is removal of the incorrect base by an appropriate DNA N-glycosylase to create an AP site. This is followed by nicking of the damaged DNA strand by AP endonuclease upstream of the AP site, thus creating a 3' OH terminus adjacent to the AP site. The third step involves extension of the 3' OH terminus by a DNA polymerase accompanied by excision of the AP site. Let us now move on and talk about this pathway in detail. Specificity of the various pathways is conferred by DNA and glycosylases. These are small monomeric enzymes with no cofactor or divalent cation requirement. Most are highly specific for a certain type of altered base, but some have more relaxed specificity. The crystal structures of many of the DNA glycosylases have been determined. They are similar to each other and they suggest a common mode of action with variations depending on specific structure recognized by the glycosylase. It appears that DNA glycosylases recognize the damaged base and gently pinch the DNA while scanning it, with the result that the DNA kinks at positions of instability caused by mismatching. The glycosylases all possess attractive binding sites for the bases that they are designed to recognize. The DNA kinking combined with additional pushing by the enzyme encourages the mismatched bases to flip out of the DNA double helix and enter the binding site. If the binding site offers a good fit, the base remains inside long enough for its bond to deoxyribose in the DNA backbone to be severed by the enzyme. Thus, it removes the damaged DNA base. These enzymes hydrolyze the N-glycosylic bond between the base and the deoxyribose by the action of, say, in this example, uracil DNA N-glycosylase. This gives rise to an abasic site or an AP site. If the base that has been removed is a purine, it's called a purinic site. If the base that has been removed is a pyrimidine, it is called a pyrimidinic site. Otherwise, we simply prefer to call it an AP site. The AP site created by a DNA N glycosylase is identical to that created by spontaneous DNA purination or depyrimidination. The next step in BER, that is BER, is catalyzed by an AP endonuclease. These enzymes break the phosphodiester bond, making a 3' OH end for DNA polymerase. All the known AP endonucleases are categorized in 4 classes. Class 1 AP endonucleases make a 3' incision such that 3' OH and 5' phosphate ends are generated, whereas class 2 AP endonucleases make a 5' incision such that 3' OH and 5' phosphate ends are generated. Class 3 AP endonucleases are known to create 3' phosphate and 5' OH at the 3' side of the AP endonuclease, whereas class 4 AP endonucleases create a 5' incision such that 3' phosphate and 5' OH non-ligatable ends are created. The most common AP endonuclease activities are of class 2, accounting for about 90% of the total AP endonuclease in the E. coli cells. Similar enzymes are also found in eukaryotic organisms. In Saccharomyces cerevisiae, the major AP endonuclease is related to endo-4, but in mammalian cells, AP endonucleases related to exonuclease 3 are more abundant. Let us now have a bird's eye view of the events in BER up to the AP endonuclease action. After strand cleavage by AP endonuclease, the upper strand on the left side of the nick retains a 3' OH terminus that can easily be extended by a polymerase. The strand on the right side of the nick retains a 2' deoxy 5' phosphate terminus that must be removed to permit ligation. This non-nucleotide terminus is removed by one of the two alternative pathways which we will see. In the mammalian cells, the major pathway is mediated by DNA polymerase beta, which has two distinct enzymatic activities. Using its polymerase activity, polymerase beta incorporates the correct nucleotide at the AP site, in this case C, and then with a recently discovered deoxyribose phosphatase activity or DRPase activity, it excises the deoxyribose phosphate moiety. This pathway, utilizing DNA polymerase beta, generates a repair patch, only a single nucleotide in length. The final ligation step is carried out by DNA ligase in partnership with the XRCC1 protein. The alternative longer patch pathway is also sometimes employed in mammalian cells and is usually employed in yeast cells. The longer patch pathway depends on enzymes normally involved in DNA replication. The mechanism probably involves synthesis of a new stretch of DNA several nucleotides long by DNA polymerase delta or epsilon. This results in displacement of the strand terminated by the deoxyribose phosphate group. The displaced strand is then probably removed by a structure-specific nuclease, particularly F1 endonuclease, to create a ligatable substrate. This enzyme appears to play a similar role in the maturation of Okazaki fragments behind replication forks. Finally, DNA ligase 1 seals the nick and the damage is repaired. We will be talking about mismatch repair pathway followed by the SOS response. Mismatch repair pathway, a pathway completely dedicated to repairing the mismatches that happen at the time of replication. The action of DNA polymerase 3, including its proofreading activity, results in one misincorporation per 10 to the power 8 bases synthesized. Some mismatches due to replication errors escape proofreading. Mismatch repair reduces this rate to one chain in every 10 to the power 10 or 10 to the power 11 bases, thus increasing the accuracy of DNA synthesis by 2-3 times. MMR recognizes mismatched bases in DNA, for example, G against a T or A against a C base pair. These do not cause large distortions in the helix. The mismatch repair system apparently reads the sequences of the bases in the DNA. Most mismatches are due to errors during replication. However, mismatches can also be produced by other mechanisms, for example, by deamination of 5-methylcytosine to produce thymidine, improperly base paired to G. Regardless of the mechanism by which they are produced, mismatches can always be repaired by the mismatch repair system. This repair system has to work quickly, otherwise, the mutation would get fixed and the repair system would not have any idea about which was the mispair. For example, this is a double-stranded DNA molecule which is undergoing its first round of replication, and during replication in one of the duplexes, you can see here that against an a G was incorporated. So this is the mispair here. In the second round of replication, against this G, a C would be added. And now the repair system would have no idea which of them is the wrong base pair that has been incorporated. We say that after the second round of replication, the mutation has been generated or the mutation has been fixed. Hence, the mismatch repair system has to work before the second round of replication, which means that before the mutation has been fixed. The mechanism of mismatch repair system was first thoroughly studied in E. coli. The proteins that initiate the repair process are MUT-S, MUT-L, and MUT-H. MUT-S is responsible for initiation of E. coli mismatch repair. It is a 95 kilodalton polypeptide which exists as an equilibrium mixture of dimers and tetramers and recognizes mismatched base pairs. MUT-L, a 68 kilodalton polypeptide that is dimeric in solution, is recruited to the heteroduplex in a MUT-S and ATP dependent fashion. MUT-S, MUT-L heterocomplex is believed to be a key intermediate in the initiation of the mismatch repair. MUT-H is the endonuclease that brings about the NER. In the figure, the replication error produced mismatch is indicated by the distorted double helix. You can see that the distortion here is because of the mismatch that has occurred during replication. MUT-S scans the newly replicated DNA and recognizes such mismatches due to the distortion they cause in the DNA backbone. The mismatches are true mismatches plus loops caused by insertions or deletions of up to 4 nucleotides. And it binds to the mismatch containing DNA, inducing a pronounced kick in the DNA and a conformational change in the MUT-S itself. The next step is where MUT-L now binds to the MUT-S DNA and stabilizes the complex. The MUT-S MUT-L complex activates the next enzyme MUT-H to come to the site, which binds to the parental strand. It is a U-shaped enzyme which sits on the parental strand and crosses its arms so as to cause a nick in the daughter strand. The nick can be introduced either on the 5' side or the 3' side of the mismatch. Depending on the nick site, whether it is 3' or 5', different exonuclease is recruited and it is responsible for removing a few nucleotides along with the mismatch. As a result, a gap is created which is filled by DNA polymerase, which binds to the 3' OH end and uses the parental strand as template to synthesize the DNA. Once the gap is filled, the nick is sealed by ligase and the mismatch is repaired. The major challenge faced by MMR is to correctly recognize the newly synthesized daughter strand that has the mistake and leave the parental strand alone. How does E. coli mismatch repair system know which of the two mismatched nucleotide is to be replaced? At least in E. coli, this is not a problem. This is because E. coli genome contains palindromic GATC sequences every 250 base pairs. The adenines in these GATC sites are methylated. GATC is a palindrome, so the opposite strand also reads GATC in the 5' 3' direction. This means that a newly synthesized strand across from a methylated GATC is also destined to become methylated. This methylation is brought about by DAM methylase, which closely follows the newly replicated strand. It takes approximately 1 minute to reach the methylation sites. For this 1 minute, the parental strand is methylated and the daughter strand is non-methylated. E. coli MMR exploits this difference in the two strands to correctly recognize the daughter strand from the parental strand and correct the mismatch in the daughter strand. This process must occur fairly soon after the mismatch is created, or both strands will become methylated and no distinction then will remain between them. E. coli DNA is normally methylated at GATC sites, just like I told you now, but the newly synthesized strand is not immediately methylated. The fact that the old strand but not the new is methylated near the replication fork allows E. coli cells to distinguish the old strand from the newly synthesized daughter strand, which has the mispair. We have seen that different exonucleases can be used to remove the single-stranded DNA between the nick, which has been created, the mute H, and the mismatch. Mute S recognizes the mispair and mute L stabilizes it. Mute H binds to the nearest methylated adenine on the parental strand. We have seen this just now. If the nearest methylated adenine is on the 5' side of the mispair, mute H brings about a nick on the 5' side of the mispair. In this case, a 5' 3' exonuclease is recruited to remove the oligonucleotide along with the mispair. DNA polymerase binds to the 3' OH end, fills the gap, and the ligase seals the nick. On the other hand, if the nearest methylated adenine is on the 3' side of the mispair, mute H brings about a nick on the 3' side of the mispair. In this case, a 3' 5' exonuclease is recruited to remove the oligonucleotide along with the mispair. DNA polymerase then binds to the 3' OH end, fills the gap, and the ligase seals the nick. It is important to note that the use of methylation to distinguish the parental strand is probably peculiar to E. coli. Data from yeast and vertebrate in vitro mismatch repair experiments suggest that single-strand nicks provide a signal for strand specificity in these organisms. Note that single-strand breaks are present in nascent DNA strands between Okazaki fragments in the lagging strand and at the 3' end of the leading strand. Thus, this method of recognizing the daughter strand from the parental strand by methylation is present in prokaryotes, but it is absent in eukaryotes. Eukaryotes recognize the daughter strand from the parental strand because of the fact that the daughter strand, the lagging strand, is formed in the form of Okazaki fragments, and there are several nicks present in the daughter strand. Human homologs to mute L and mute S have been discovered because mutations in them can cause one of the most common hereditary cancers, the hereditary non-polyposis colon cancer. Whereas mute S and mute L function as homodimers, the eukaryotic proteins function as heterodimers. Heterodimers of mute S homologs are responsible for initial recognition of the mismatches and small insertions or deletions, and heterodimers of mute L homologs interact with the resulting complex, as in the case of E. coli. In contrast to E. coli, however, some eukaryotic mute L homologs play a specific catalytic role in mismatch repair. The eukaryotic mute L homologs have an important endonuclease activity. Two heterodimers of mute S homologs are found in human cells. One of these dimers is called mute S alpha and the second is called mut-S beta. The first heterodimer preferentially recognizes single base mismatches and small incision deletion loops of up to 1 to 2 bases. The second heterodimer primarily recognizes larger loops of 2 to 10 bases. Most, that is almost 80 to 90% of the MSH2 in the cell is complexed with MSH6 to form mut-S alpha. Thus, mut-S beta is a relatively minor activity. Structural studies of mut-S homologs suggest that MSH2 resembles the B monomer of the mut-S homodimer, while MSH3 and MSH6 resemble the A monomer. Three mut-L homolog heterodimers are known. MLH1 is one of the dimer subunits in every case. The major heterodimer, mut-L alpha, consumes about 90% of MLH1 and contains PMS1 in addition to MLH1. The second heterodimer, mut-L beta, consists of MLH1 and MLH2 in yeast as well as in humans. Note that human PMS2 is a better homologue of yeast PMS1 than is human PMS1. The third dimer, mut-L gamma, consists of MLH1 and MLH3. The first dimer plays a major role in mismatch repair. The role of mut-L beta is not clear. Mut-L gamma plays a minor role in mismatch repair system. Thus, in today's lecture, we have talked about mismatch repair system in prokaryotes, taking E. coli as the model organism. And we have seen how the E. coli mismatch repair system recognizes the mismatches and repairs it using the method of methylation to recognize the daughter strand from the parental strand. The eukaryotic mismatch repair system is slightly complex with heterodimers present in the instead of monodimers, and this particular mismatch repair system is responsible for removing the mismatches that have been created during the replication process. We are going to talk about yet another repair system, that is SOS response, which is also called the translation DNA synthesis or the error-prone bypass system. In all the pathways that we have studied in the previous video lectures, damage to DNA is mended by excision, followed by resynthesis using an undamaged template. But at times, a replicating DNA polymerase encounters a lesion such as a pyrimidine dimer or an apurinic site that has not been repaired. Because such lesions are obstacles to the progression of the DNA polymerase, the replication machinery is forced to stop, or it must attempt to bypass and copy across the lesion. One such mechanism of bypassing the damage is translation DNA synthesis. Although this mechanism is highly error prone and thus likely to introduce mutations, translation DNA synthesis works on the principle that to survive with mutations is better than to perish or die. Because if the DNA replication is not completed, the cell will face the worst fate, that is death. DNA repair pathways can subsequently later on correct the damage. Translation DNA synthesis is catalyzed by a specialized class of DNA polymerases that synthesize DNA directly across the site of the damage. In E. coli, DNA polymerase 4 or DNA polymerase 5 performs translation DNA synthesis. An important feature of these polymerases is that although they are template dependent, they incorporate nucleotides in a manner that is independent of base pairing. This explains how the enzymes can synthesize DNA over a lesion on the template strand. But because the enzyme is not reading the sequence information from the template, translation DNA synthesis is often considered to be highly error prone. Because of its high error rate, translation DNA synthesis is actually considered to be a system of last resort. If the cell has no other repair system as an option, it will consider the bypass mechanism. It enables the cell to survive what might otherwise be a block to replication, but the price that is paid is a higher level of mutagenesis. For this reason, translation DNA polymerases must be tightly regulated. In E. coli, the translation polymerases are not present under normal circumstances. Rather, their synthesis is induced only in response to DNA damage. Thus, the genes encoding the translation polymerases are expressed as a part of a pathway known as the SOS response. SOS repair occurs when cells are overwhelmed by UV damage. This allows the cell to survive but at the cost of mutagenesis. SOS response only triggers when other repair systems are overwhelmed by the amount of damage so that unrepaired DNA accumulates in the cell. SOS response, which is also called save our ship or translation DNA synthesis, incorporates random nucleotides leading almost always to mutations and, like I have just now said, therefore is also called the error-prone repair mechanism. Let us further discuss this error-prone repair mechanism. The error-prone repair mechanism involves DNA polymerase and two other genes products encoded by the UMU-CD operon. The UMU-CD proteins are produced in times of dire emergency and instruct the DNA polymerase to insert any bases opposite the thymine dimers as the DNA damage would otherwise be lethal. The risk of several mutations is worth the risk as measured against the threat of death. The question is, how is this SOS repair activated? In response to extensive DNA damage, there is a regulatory system that coordinates the bacterial cell response. This results in the increased expression of more than 30 genes involved in DNA repair. These include REC-A, which is the activator of the SOS response, recombination SFI-A, a cell division inhibitor, which includes SUL-A and SUL-B activated by SOS system, and these inhibit cell division in order to increase the amount of time the cell has to repair the damage before replication. UMUC and D, an error-prone bypass of thymine dimers, and the SOS system or the SOS response is very tightly regulated by two important genes, REC-A and LEX-A. LEX-A normally represses about 18 genes. The SOS response box, as it is called, includes LEX-A, REC-A, UVR-A, UVR-B, UVR-C, UMU-C and D, SUL-A, SUL-B, and SSB. Each gene has an SOS box in the promoter. LEX-A binds to this SOS box to repress the expression of all these genes. The SOS response is induced by DNA damage, including the ultraviolet damage, and depends on the product of the REC-A gene, like I said just now. Ultraviolet light or another mutagenic treatment somehow activates the REC-A coprotease activity. REC-A is a very versatile enzyme and protein, and it also has this coprotease activity. This coprotease has several targets. One is the lambda repressor, but its main target is the product of the LEX-A gene. This product, LEX-A protein, is a repressor for many genes, including repair genes. When it is stimulated by REC-A coprotease, to cleave itself, all these genes are then induced. Two of the newly induced genes are UMUC and UMUD, which make up a single operon UMUCD. The product of the UMUD gene is clipped by a protease to form an active form of UMUD, which associates with the UMUC, and together it forms a complex. This complex has DNA polymerase activity, so it is also referred to as the DNA polymerase 5. DNA pol 5 can cause error-prone bypass of pyrimidine dimers in vitro on its own, but polymerase 3 holoenzyme and REC-A protein stimulate this process considerably. Such bypass involves replication of DNA across from the pyrimidine dimer, even though correct reading of the defective strand is impossible. This avoids leaving a gap, but it frequently puts wrong bases into the new DNA strand and hence the name error-prone. Once damage is repaired, REC-A no longer catalyzes the cleavage of LEX-A, which is actually still being made. So uncleaved LEX-A accumulates and it turns the SOS response system off again. Let us see what we have just now studied. This is a DNA polymerase 3, which is replicating and just ahead of the pyrimidine dimer, which causes a distortion. It has stopped because it cannot pass the lesion here. Okay, at this point, this DNA polymerase 3 will dissociate and its place will be taken up by DNA polymerase 4 or 5, which has just now been formed as we have seen in the previous slide. This polymerase 5 will bind and it will add any two bases against the pyrimidine dimer. As you can see in this slide here, against a T, a G has been added here. This polymerase 5 will add these two bases and it will fall off the template because it is not processive. It is again, the 3' OH end is again loaded by DNA polymerase 3 here, which then continues the replication. So, DNA polymerase 3 with the help of DNA polymerase 5 has been able to carry out what is called as translation DNA synthesis. Let us now talk about the error-free and error-prone bypass in humans. Do the humans also have such an error-free bypass system? Human cells also have an error-prone system to bypass pyrimidine dimers. It involves a distinct DNA polymerase known as DNA polymerase zeta that inserts bases at random to get past the pyrimidine dimer. But human cells also have a relatively error-free bypass system that automatically inserts two DAMPs into the DNA strand across the pyrimidine dimer. Thus, even though the bases in the dimer cannot base pair, this system is able to make the correct choice if both bases in the dimer are thymines, which is often the case. The DNA polymerase involved in this relatively error-free bypass system is called the DNA polymerase eta. It was discovered that the defective gene in the patients with the variant form of XP, that is xeroderma pigmentosum, is the gene that codes for the DNA polymerase eta. Thus, these patients cannot carry out the comparatively error-free bypass of pyrimidine dimers catalyzed by DNA polymerase eta and therefore have to rely on the error-prone bypass which is catalyzed by DNA polymerase zeta. This error-prone system introduces mutations during replication of pyrimidine dimers not removed by the excision repair system. However, because these patients have normal excision repair, few dimers are left for the error-prone system to deal with. Although the term error-free for DNA polymerase eta is justified in terms of its ability to deal with thymine dimers, it actually is a misnomer. When the fidelity of this enzyme was tested in vitro using a double-stranded DNA with a gap in it, it was found that DNA polymerase eta had a lower fidelity than any other template-dependent DNA polymerase ever studied. One mistake per 18 to 380 nucleotides incorporated. Thus, it is a good thing that cells normally have the nucleotide excision repair system. Without it, DNA polymerase eta would be a very poor backup for dealing with anything but thymine dimers. As compared to it, DNA polymerase zeta is 20 times more accurate than eta in its polymerizing activity except against the thymine dimers. If DNA polymerase eta gene is defective, DNA polymerase zeta and others take over. They cannot carry out the comparatively error-free bypass of pyrimidine dimers, which is catalyzed by DNA polymerase eta, and as a result of that, introduces mutations during replication of pyrimidine dimers. Let us try and understand all this that we have talked about just now with the help of this figure. So here in humans, you can see that this is our replicative DNA polymerase, that is DNA polymerase delta, if you remember well, and it is replicating across the pyrimidine dimer. When it comes across the pyrimidine dimer, it will fall off the template DNA. At this point, DNA polymerase eta will take over. Eta will add two A's against the two T's. So here you can see that DNA polymerase eta, which is shown in this blue color here, has added two A's here against T's. This it is doing without actually reading the template. And after it has added these two A's, it will fall off. And this 3' OH end is again free for DNA polymerase delta to bind and to continue the replication. This is the error-free system in humans, or the error-free bypass system in humans. Now, like I said, this is DNA polymerase delta, which is carrying on the replication. And when it comes against the pyrimidine dimer, it will stop here. It will dissociate and its place will be taken by DNA polymerase eta, which will add two A's against two T's. And as a result of that, it is called as the error-free bypass system. But in case the cell doesn't have the DNA polymerase eta, then the cell has to depend upon the DNA polymerase zeta, which is responsible for the error-prone bypass. DNA polymerase zeta invariably adds any two nucleotides. So it can add any two nucleotides against this pyrimidine dimer and it will move ahead. And then DNA polymerase delta will take over the replication. But because here any two nucleotides have been added against the pyrimidine dimer, it is going to lead to mutations. And that is why this system is called the error-prone DNA bypass system. I hope you people have understood it. So in humans, actually what happens is that, let us understand this again because it is slightly complicated. This yellow one is my replicative polymerase, that is the DNA polymerase delta. So it is replicating, and if you remember well, it has a pCNA which helps to make it processive. So this DNA polymerase delta is replicating the strand, and when it comes against this DNA lesion, it will stop. Once it stops, it will fall off and it will be taken up by DNA polymerase eta. Now remember, DNA polymerase eta is responsible for the error-free bypass. So this eta will add two A's against the two T's and bring about the translation DNA synthesis. But the problem with this DNA polymerase eta is that if it falls off from here right now after adding two A's, then this DNA polymerase delta cannot bind to the 3' OH end here because DNA polymerase delta cannot bind to an unpaired nucleotide with a 3' OH end. So this means that even if DNA polymerase eta falls off from here, delta cannot come and bind. In this case, what happens is that after eta has left the site, DNA polymerase zeta takes over because it has the capability of binding to a 3' OH end of an unpaired nucleotide. Now zeta, if you remember, in the previous slide I have told you just now, it is 20 times more accurate than eta, except the pyrimidine dimer. But the pyrimidine dimer part has already been dealt with by DNA polymerase eta. So now this zeta will add a few nucleotides, and most of the times the nucleotides added by zeta are correct. Because it is not processive, it will fall off the template DNA, and now DNA polymerase delta is capable of binding to the 3' OH end of a paired nucleotide. And then DNA polymerase delta will carry on the replication, and in this way this damage is still there, but it has been bypassed by these three polymerases taken together. So humans have two systems. They have DNA polymerase eta for error-free bypass, and if it is not present, they will have to depend on DNA polymerase zeta for the error-prone bypass. But in the cells which have both the polymerases, this is the system which is followed, and this is the way in which this translation DNA synthesis takes place in the human cells. With this, we come to an end of this lecture, where we have mainly today talked about the SOS response, how it is triggered, how it is expressed in both the prokaryotes and eukaryotes. And if you like the content of this channel, please take some time out to like, share, and subscribe, and do comment if you have any queries regarding to any of the topics that have been discussed. Thank you.